In this two-part video, I will describe the individual slides of bronchiogenic carcinomas. Now, remember that these tumors originate from the epithelium lining the respiratory tree and represent the vast majority of lung cancers, but it excludes such things as mesotheliomas that arise from the outside linings of the lungs and metastases of other cancers to the lungs. Now, before you look at the lung tumors, I want to briefly review the normal architecture of the lungs. Perhaps the best way to understand the structure of the lungs is to follow the air passageways. Here you can see the trachea coming and dividing into two primary bronchi. One goes to the right and the other to the left lung. They enter the lungs at the hilus, which is also where the blood vessels and nerve enter and exit. Now you can visualize the airways of the lungs as branching trees. The bronchi are the largest air passageways and are characterized by the presence of cartilage in their walls and are lined by pseudostratified columnar epithelium with goblet cells and cilia, which is also known as respiratory epithelium. After branching and narrowing, the bronchi give rise to bronchioles, which lack cartilage in their walls, and are lined by epithelium that transitions from pseudostratified columnar to simple cuboidal. And finally, the bronchioles branch and narrow further and end in alveoli that are lined by a squamous epithelium that allows gas exchange to take place. I would like you to start out by examining slide number 97 of the normal monkey lung. However, there are several features of this section of which you should be aware. First, the section has been perfused. That is, fixative has been forced through the blood vessels, replacing the blood. Secondly, fixative has also been forced through the trachea so that the lungs are inflated. Now, this is not the case with the pathological specimens in which the lung tissue is collapsed. And thirdly, this section is very old and much of the hematoxylin staining has faded. Now place the slide on a piece of white paper and observe that the section is light staining and consists of a highly branched network. The arrow indicates the region of the hilus that contains the large airways and blood vessels that I want you to observe first. Center this region on the microscope stage and examine it under low power. This image shows a large blood vessel labeled BV, and you can still see some of the residual blood in the lumen. Below and to the right is a bronchus, which can be identified by the piece of cartilage labeled C here in its wall. Now, in this section, the cartilage stains eosinophilic, but in other sections, it will be very basophilic. Note that the cartilage has a slight curve to it, and the concave surface is next to a bronchus that is lined by a thick respiratory epithelium. The convex surface of the cartilage is next to a bronchiole that is on the left here. Remember, cartilage is found in the walls of the bronchi, but not the bronchioles. However, in this particular case, the bronchus just happens to be located next to a bronchiole. Center the field on the epithelium lining the bronchus, that is, on the concave side of the cartilage, and switch to higher power. Note that the epithelial cells are columnar and have cilia, which appear as fine filaments on their free surface that help to move the mucus. This is pseudostratified, ciliated columnar epithelium with goblet cells, which is also known as respiratory epithelium. Next, examine the bronchiole that is on the convex side of the cartilage, that is on the left, under higher magnification. Follow the respiratory epithelium along and observe that the cells transition from a thick columnar to a simple, that is a single, cuboidal layer. These cuboidal cells include both ciliated cells and the non-ciliated Clara cells that secrete mucin. Finally, examine the tissue outside of this region that contains the alveolar ducts and sacs where gas exchange takes place. The epithelium here is thin and contains capillaries. However, these are empty of blood in this particular section. Now, I would like to remind you about the classification of bronchiogenic carcinomas as shown in this diagram. Remember that they are classified according to their morphology and response to treatment into two major categories: small cell carcinomas and non-small cell carcinomas. And the non-small cell carcinomas can be further divided into three subcategories: squamous cell carcinoma, adenocarcinoma, and finally large cell carcinoma. Now, this figure is taken from a pathology textbook and shows a side-by-side comparison of these four different types of bronchiogenic carcinomas. In the upper left quadrant is a sample of small cell carcinoma, and as the name implies, the cells are small and have compact nuclei. Below this is an example of squamous cell carcinoma. Basically, these cells resemble those that give rise to squamous epithelium, such as that of the skin. Well-differentiated forms of these tumors often display distinct desmosomal junctions between the cells and can form keratin pearls as shown here. In the lower right quadrant is adenocarcinoma, which has a gland-like appearance and may contain mucin in their lumens. And finally, in the upper right-hand quadrant is large cell carcinoma. This is also known as undifferentiated lung cancer because of its anaplastic nature. This is a diagnosis of exclusion and probably represents squamous cell or adenocarcinomas that are so poorly differentiated that their origins cannot be determined. Now, the first bronchiogenic carcinoma that you will examine is an example of small cell carcinoma. This figure illustrates how this sample was obtained. In this case, a needle was passed through the skin into the lung and a small core biopsy was removed for analysis. However, biopsies may also be taken through the bronchus if the mass is centrally located. If diagnosed, small cell carcinoma cannot be cured by surgery because, in general, it has already metastasized. That is why we do not have a larger specimen. This form of cancer is very lethal. Place the slide labeled Lung 1 of small cell carcinoma on a piece of white paper and observe that it contains several small fragments of tissue from needle biopsies. Next, place the slide on the microscope stage and under low power, locate one of the fragments and switch to higher power. Observe that most of the nuclei are dark staining and are generally round. Scan the tissue for an area where you can see an entire cell and observe that it is relatively small. Indeed, they resemble lymphocytes but are somewhat larger. In some regions, you may observe tumor cells that appear to be smudged. These tumor cells are fragile and smear easily when biopsies are taken. Indeed, this is one of the diagnostic features of small cell carcinoma. Next, locate one of the larger fragments on the slide. Here you will observe that the tumor cells are arranged in clusters and are surrounded by large areas of necrosis, which is marked by large eosinophilic regions that lack nuclei. This is also a characteristic feature of these tumors. Move on now to the slide labeled Lung 2 that contains squamous cell carcinoma. Carefully examine this slide by eye and observe that it actually contains two separate fragments that are closely opposed to each other. They are separated from each other by a small gap that contains a brown staining material that was used during surgery as a disinfectant called wescodine. The center of the larger fragment contains a single tumor mass, and adjacent to this are several smaller, independent masses surrounded by collagenous connective tissue. The location of two very small pieces of cartilage that stain a dark blue is indicated by one of the arrows. Under low magnification, locate the two small pieces of cartilage. Now, situated between these two pieces of cartilage is a small region of respiratory epithelium. Examine this epithelium under high magnification and confirm that it contains cilia on its free surface. The presence of these structures indicate that this section was taken from the hilar region of the lungs where the bronchi are located. Scan the section under low power and locate a region of normal alveolar tissue on the edge of the larger fragment. Examine this under higher power and observe that there are tumor masses invading into this region. Scan the top side of the section, assuming that it is in the same orientation as shown before, under low power for a region of connective tissue that contains small black particles. Examine these particles under higher power. The black particles are soot from cigarette smoke or from air pollution that has been phagocytosed by macrophages and related cells. These cells have then migrated into the connective tissue. This type of lung cancer is closely correlated with cigarette smoking. Examine the central tumor mass in the larger fragment. Note that it consists of a highly convoluted mixture of both tumor cells and connective tissue. The boundary between these two is very sharp. The connective tissue contains fibroblast, collagen fibers, adipose tissue, lymphocytes, and smooth muscle cells. Many of the tumor masses contain central necrotic regions in which the nuclei are condensed and fragmented. Under high power, you may be able to observe small desmosomal junctions between the tumor cells, and these structures are characteristics of squamous cell carcinoma and also found in normal stratified squamous epithelium. Now, this sample has been graded as moderate to poorly differentiated, since there are no obvious keratin pearls which would be found in well-differentiated versions of squamous cell carcinoma. Now, at this point, I'm going to ask you to move on to the second part of this video entitled TBIO-572-Lung-Cancer-Lab-pt-2A.